go ahead and introduce you.
Great.
So it's my pleasure to introduce Dr. Tom Coulter. He is a I'm not even speaking now anymore. I apologise, Tom. It's my pleasure to introduce Tom Coulter, a seasoned medicinal chemist with diverse experience spanning small molecules, as well as protein-based therapeutics and drug delivery. And he's currently the Integrated Drug Discovery Director at Selvita and is focused on development of enhanced drug discovery capabilities. Tom's experience includes a 16-year tenure at Evotech, where he was SVP of Drug Discovery, as well as senior drug discovery positions at quite a few British biotechs, including Syntaxin, Caris Therapeutics, and Midotech Pharma. His achievements include management of diverse discovery programs, delivering multiple drug development candidates. So again, it's my pleasure to introduce Tom. He's going to share with us a flexible approach to small molecule drug discovery. And with that, I'll hand the floor over to you, Tom.
Excellent. Thanks very much, Karen. And thanks to Precision Globe as well for the opportunity to talk today. It's my pleasure to be here and to speak to everyone about some of the work that we've been doing recently at Selvita. Now, can everyone see my slides at the moment, or what's the status? Great. Excellent. Okay, so just for everyone's understanding, I'm, as Karen says, I'm working with Selvita. I'm Integrated Drug Discovery Director there. I'm involved in a range of projects. We're a contract service company, so I'm doing projects with clients, across the globe, and wanted to let you know about some of our experiences and learning and maybe some insight in the short presentation today. If I could get the next slide, or can I advance them myself? Okay, great. Okay, so I think one of the things I'm sure I could imagine that this kind of thing's been used quite a few times in the conference to date. You will have seen this many times before, so I'm not going to dwell on it. The key message is, we're all working in this sector, and I think we all recognize that there's still a terrible problem with attrition in drug discovery. For every approved drug, there's something over the order of thousands of compounds anyway that are behind it in early stage testing. So not every project goes all the way through to the clinic clearly, and there's a lot of things fall by the wayside. So what can we do about it? Well, there's a great industry around creating new technologies, things to help us make and find better quality drugs. But is there anything else that we can do really to enhance our chances of success? And I think one of the things that I notice as being in a CRO is many of our clients talk to us about money, resources, and time. And time is the one resource that is probably the most precious of all, because once it's gone, without the aid of a DeLorean motor car, you can't go back in time to revisit that time. And for that reason, like we at Selvita have been increasingly doing projects where we're trying to mitigate risk for failure, and things do fail. And this is really going to be the focus for the talk today. Next slide, please. New technology, new innovations, I think, are abundant. New ways to screen for compounds, and new tools to help us design problems and predict things, and artificial intelligence. There's a whole massive industry of things happening around us. But I think if you look at the actual the reasons for failure in early stage discovery, and so I'm not talking about clinical failure, and I think that this is by no means an exhaustive list, hence the white space for you to add your own issues. But failure to translate from in vitro to in vivo, it could be that despite initial validation, that once you get into animal models, that compounds just simply don't do the job they do in vitro. Also, the pharmacophore could be, for example, very lipophilic. It might demand a very lipophilic molecule. That may be very difficult to deliver. There could be other issues around it. Generally, ADME issues along the way. Once we get far enough down the track and we look at, in more detail, metabolism and things like enzyme induction, there are problems there that may be difficult to resolve or too costly and time-consuming to resolve. And of course, at any time, there's the risk that we could be gazumped by a competitor patent appearing. So these are kind of fairly typical challenges. But one of the things that, you can enhance your chances of success, and we talk about more shots on goal, by, like, for example, having a candidate and a backup that are from different structural series. But what about taking that a step further and having entities that are from different small molecule modalities? And this is something that we've been increasingly seeing in some of the projects that we've been doing. Next slide, please. Before I get into it, a couple of slides just to introduce who Selvita are. I mentioned we're a CRO. We're based in Eastern Europe. We have two main R&D facilities, one in Krakow in Poland, a small unit in Poznan, Poland, and another big one in Zagreb in Croatia. We've been going for about 13, 14 years as Selvita. Although we recently completed the acquisition of Fidelta, who's another CRO based in Croatia. So together we provide integrated drug discovery services and beyond. We've got a very strong track record and have seen quite a bit of growth over the recent years, but I think it's set to increase significantly going forwards over the next 2-3 years. Next slide, please. Key areas of expertise are indicated here. Obviously we're CROs, so we will be flexible and work in different areas, wherever the clients take us. But over time, we've built up some depth of experience in oncology, neuroscience, and respiratory disease. In Krakow and in Zagreb, there's a great depth of experience in anti-infectives, inflammation and fibrosis, and notably the Fidelta group actually has its roots in different entities in Zagreb going back over 60 years. So I think together there's collectively a tremendous depth of drug discovery experience in the company. Next slide, please. Together we're providing integrated and standalone services in preclinical drug discovery. We're going from target validation all the way through to characterization of preclinical candidates. And some of that work's multidisciplinary integrated, some of it is pure standalone. Our strengths are significant in chemistry. We are also strong in vitro and in vivo pharmacology. And we have facilities that have undergone various inspections and accreditations. So we're, like, for example, GLP and ALAC accredited. Next slide, please. We've been pretty successful. We've identified multiple preclinical candidates, and there's over 7 of them which are now in the clinic. So we're pretty pleased with that track record, and we're very much focused on delivery of value for clients. So this is it's a key measure for us to increase these totals. Next slide, please. So what I wanted to spend the time talking about is the importance of flexible drug discovery. And what I mean is, not to meander around and do kind of anything you like in a project, but not to put all your eggs in one basket. Whenever you do work on targets, there's ones that are well precedented in the literature, and it provides an easy opportunity to follow competitors or follow the literature. Targeting a catalytic site in a kinase is maybe not so unusual, but if your target is a bit more novel, validation can tell you about the importance of the target in disease, how important it is to enhance or to minimize the role of that target in a disease setting, either so that you have an impact on the disease or some of the symptoms of disease which you may wish to treat. And it'll suggest maybe how you might be able to address levels or functionality of your target, but not necessarily spell out which is the cleverest and most likely to succeed approach. So if we go on to the next slide. So let's assume we've done target validation. We've established we have a protein. We've done work around it. We understand its role in disease. We know that by suppressing the expression, for example, we may see a positive or negative correlation with increase in disease symptoms. And we then need to think how we'll actually in practice do that with the drug. And this is an example of a protein. I've indicated a catalytic site, and I've also indicated a site that's involved in a protein-protein interaction, which actually is involved in the creation of the catalytically active dimer. So it's the dimer that's active, and potentially, we can target these sites or other sites using different types of modalities. We might be able to target something upstream in a signaling cascade, or we might be able to target the catalytic site itself. And often, if there's a catalytic site, a nice small molecule binding pocket, then that's what we'll do. We'll go for that. And some of the things that we have gained experience in are you're working with traditional reversibly binding catalytic site inhibitors, but also protein-protein interactions. Recently, we've done quite some work in the area of PROTACs, and we're also seeing a great increase in the field of interest in targeted covalent inhibitors. So these are all like different things, quite different things, that offer you maybe more diversity than you'll get from getting two types of chemical series that are both reversible catalytic inhibitors, for example. So next slide, please. One of the first things that I want to talk about, I think that so much work in drug discoveries has been focused on small reversible molecules. I'm not going to dwell on that in this short presentation, but I wanted to look at like the difference between small molecules versus PROTACs, and clearly with a small molecule, you're designing an agent to go into a pocket, bind, and inhibit, one molecule of the target protein, for example, and you'll need to use an excess of your inhibitor generally to drive the occupancy to a level that provides a useful therapeutic effect. This can be complicated because sometimes molecules don't bind as well as we would like them to, even in a catalytic pocket. And clearly there's also issues with resistance. If the protein tends to undergo mutation in key parts of that binding pocket, it can mean that your compounds are very quickly taken care of in vitro or in vivo, let's say, and you need to find another molecule. PROTACs are very interesting. I'm sure lots of you are working in this field or have experience in it. But I think that it's very nice because you have a catalytic action of the PROTAC, typically. You don't necessarily need to bind in a catalytic pocket, although you can do that if you choose to. You can use a different site on the protein altogether, and it doesn't necessarily need to be one that has a functional role. It opens up opportunities, especially if you run into challenges with how your small reversible inhibitor binds. And I think one other key consideration here with PROTACs is that often the interaction between the PROTAC and the protein to be degraded doesn't need to be one that is optimized to a high degree of affinity. So if you've got something that's like 1 micromolar binder or better, then you're in good shape to potentially see that degraded with a good PROTAC. Next slide. At Selvita, we've been working on PROTACs over the past 3 years, and we've gained quite a bit of experience, and it's ranged from projects where we've been making components for PROTACs, building blocks, or functionalized entities that are incorporated into PROTACs, or we've been at the other end of the spectrum doing the design, synthesis, and the biological and chemical characterization of PROTACs. So it's the full spectrum of experience. I think it's been very useful. Right now, there's something like a quarter of the chemistry team has had some experience in this field. So it's definitely one where we see there's a lot of demand, and where we've now been able to develop a great critical mass of knowledge and experience in this field. Next slide, please. There's a few key elements, I think, that really underpin the success on a PROTAC project. One is the chemistry, medicinal chemistry, and it would include the computational chemistry around PROTACs. Another is clearly the biological characterization of the PROTAC. And for us, our experience in the area has been enhanced by the diversity of work that was done before we started working on PROTACs. So there's a lot of work that we've done where we generated other types of drug conjugates, and where we worked with ligands for nanotechnology applications and stuff like that. So there's very much a range of different experiences that have come together into one solid strategy for PROTACs. And we've done a lot of work optimizing the binding of small molecule ligands to protein targets. That's very much important in terms of getting a ligand for your protein to be degraded, and also a ligand for the ligase. And the other area that we've had a lot of experience and learned a lot in is in terms of the purification and characterization of these things. They tend to be a lot bigger than the sort of things that people who are used to Lipinski rules are working with. And very much it's been a learning experience for people. Some of the chemistry that we've done that looks very simple on paper, in practice becomes very low yielding and strangely complicated. And we've got a lot of insight into that now. And I think we understand, what pitfalls to avoid and how to maximize chances of success on these things. Next slide, please. We've worked through all the stages of producing the PROTAC, selection of the ligase, identifying ligands for the ligase and the target protein, incorporation of a linker into the ligands is something that can completely disrupt the binding. But we've got good strategies around all of this now. And on the next slide, you'll see that the biological characterization. Next slide, please. The biological characterization of these things is very well established with, what I'd say are very robust assays. There's nothing strange or startling here. Next slide. So I think being successful in this area depends on having the right assays and the right chemistry in place, and hopefully having the right know-how. And I think that it's certainly something that we've established. But next slide. This takes us on to the second thing that I wanted to talk about, targeted covalent inhibitors. And it's something that really everybody, I think, has been avoiding for probably 2 decades or more. Most people working in drug discovery. And covalent inhibitors are obviously use a reactive group in the molecule. It's something that is part of the PAINS criteria for assay interference. And typically these things would be thrown out and not screened. But there have been a lot of successes with covalent compounds. In the not too distant past, afatinib was approved for non-small cell lung cancer. And I think this should really signal that these things can have a good application, and that you can modulate the reactivity in a way that allows these molecules to be safe and still very efficacious. Next slide. Just a snapshot of a few of the key covalently binding compounds that have been discovered in the past 100 or more years. And I think you're looking at the names of these things, I think you would agree that these are typically what you would call blockbuster molecules. They're not insignificant compounds or niche things. So I think we really shouldn't be afraid of covalency. And nowadays, we're designing it rather than discovering it by serendipity. But it's definitely an area that where I think that there's a lot of gold to be found. And I look forward to continuing our journey that we're doing now at Selvita. Next slide. Some of the things that we've developed in terms of experience and know-how are in the areas of CNS acting targeted covalent inhibitors and also peripherally acting covalent inhibitors. Some of the design experience comes from the PROTAC area and also from nanotechnology experience. And we're very much using computational design to help us in this kind of technology. And we've been doing both screening approaches and also fast follower approaches in terms of advancing molecules. Next slide, please. We have utilized capabilities from the proteomics field, I guess, to facilitate analysis of covalently modified proteins. And this is something that is working very well in our analytical labs. Next slide, please. But I think the area where I wanted to get to is really the important thing is that if you can approach a target by using different modalities, you've not only mitigated the risk in the project, but you've also even increased the likelihood that you'll have two assets rather than one. And whenever you consider things like the properties, actually I've noticed the non-covalent and targeted covalent should be swapped around, but with targeted covalent and PROTACs, there's a potential for high potency, low clinical dose, long durations of action because you're inactivating or degrading the protein altogether. You've got to wait for resynthesis to get the functionality back. And but with normal non-covalent binders, that's where we've got most of our experience, and we know about the drug delivery, the DMPK, PKPD, etc. And the development of this kind of thing is well mapped out. But right in the middle, there's shared attributes in all of these things, and if you're using the same binding pocket to for your PROTAC to bind a target protein, or that's where you're binding your targeted covalent inhibitor, there's a lot of information that we can share working on two strategies at once about protein ligand binding and the general pharmacophore. So next slide. Clearly, whatever you do, however you go about it, it's key that you optimize your designs, test analysis, and design synthesis, testing and analysis cycles, and that's something that we very much do in-house. And the key thing is to be able to crank this wheel as quickly as you can, so that you learn which of your approaches is not going to be likely to succeed in the longer term. And if you are working through this combined kind of strategy, then you're not left at square one or with nothing. You'll have an advanced stage that you can spring from to find something that will ultimately make the project successful. And I think that we do it in screening. We'll use HTS and fragments and maybe a DEL approach. We'll use a de novo design approach using virtual screening and maybe artificial intelligence. So that diversity in finding chemical start points, I think it's important that we use that in moving things forward for the to the end of the project. And that's where I would like to round things off, and I'd be happy to take any questions.
Thank you, Tom, for your great insightful presentation and showing us an overview, most importantly, of your labs overseas in Poland. I think there may have been a comment. Sorry, it was for something else. If there's any questions or comments for Tom, you can type it here. You can also connect with his team, Andreas, via our meeting scheduler link to learn more about Selvita's capabilities. And Karen, if you have any comment.
Yeah, I have a couple of questions, and first a comment. That was a great presentation, Tom. I thoroughly enjoyed it. I think you touched on a few really important points in there, and the questions I have are really, the kind of message you gave about people having, either an additional molecule or a backup molecule that's within a slightly different series, but essentially the same overall idea, and then these maybe different modalities or different starting points, and seeing a little bit more of that. At what point do you think it might be true that actually that is the majority of what people try as their path to success versus what people have been doing for, the last few decades?
Yeah, I think that people are, without a doubt, more open to explore these different kinds of approaches. I think that what probably is happening or has happened is that they've explored a traditional approach and run into problems, and then they'll try something else. I think that there's a real benefit to doing it all together. Clearly, you've got to weigh up the costs. You've got to decide, do you really want to take both of these approaches through to different candidates? And it could be valuable because if you can end up with two entities that address different aspects of the disease, in terms of duration of action, the mode of action, resistance to disease or to resistance mechanisms. And I think that when we go forward in the future, I think that clearly we need to learn how to do these things faster and cheaper, but there will be more parallelization, I would say, of approaches.
Right. Thank you. I have one more question, and then I think we have at least a question in the chat. So you mentioned that we collectively have the most experience with the sort of non-covalent molecules versus the covalent molecules that are now being investigated, and obviously that we don't have as much understanding of the properties of those molecules and how they behave. And so the question I have for you is, is this just a matter of time to understand how they behave, or is it going to be harder to understand how they actually behave?
I think there's a lot of subtleties. I think with covalent inhibitors, it's not a matter of having a reactive group and that, if you throw it into an NMR tube with a nucleophile, that you'll see an adduct being formed, even if it's like a reasonable mimic for the actual in-cell process. I think that in some cases, at least, the protein that you bind to almost facilitates the covalent binding happening as a second step. It's not like you suddenly you've got these things flying about and then bang, there's this volatile reaction between them. It's not like that at all. I think you can get things that don't appear to be reactive in vitro, but whenever you get them into a cellular system, you do see adducts being formed. And I think that we've got a lot to learn about that. And I think, in terms of safety profile, clearly it's essential that with a covalent inhibitor that you're sure that it's not hitting something else, that you're not going to deplete enzymes in your liver in some way. Mm-hmm. So I think we're finding our way probably collectively with these things. But I've no doubt that, I've seen the interest absolutely surging on these kinds of things, and I've no doubt that we'll hear more success stories coming out in time to come.
And there is one more question for you from Miss Kinesia. Sorry if I'm mispronouncing your name. She's asking, why is there an avoidance of using covalent binding inhibitors in the pharma industry?
I think that, well, typically people have been concerned about getting false positives, about things that are very promiscuous, very reactive, and anything that is kind of remotely electrophilic. I think people have really got a fear of, and I think it's not well justified. Most chemists at some stage will have done a Michael addition. It's a pretty straightforward reaction. But if you take an unsaturated amide or an unsaturated ester, it's not going to react with everything. And I think that there is a balance of reactivity and pharmacophoric fit that I think is important to establish in these molecules. But if you have that, then I think that you're on safer ground. And I think we've seen the precedent in these compounds that I showed from the approved drugs. But yeah, I think that anything that's likely to cause a promiscuous reaction to turn up in different screens, people have kind of really screened these out. And I think in some cases it's been throwing the baby out with the bathwater,
Oh, great. Again, like I said, if you're interested in connecting with Tom, you can connect with his colleague Andreas, or you can shoot me an email. Appreciate, again, your time and your presentation, Tom.
You're very welcome.
And
thank you.
Karen, I guess we'll be moving on to our next presentation, which is Target Hit Discovery by Dr. Tabanu. So we can all head over to the